Good morning. In our last class, we were discussing the process of recombinant DNA technology, and we had already discussed the 4 steps, that is the isolation of DNA, fragmentation of DNA by restriction endonuclease, then the desired DNA fragments were isolated by using the gel electrophoresis, and by using the DNA ligase enzyme, the gene of interest was ligated into vector, and then the multiple copies of gene of interest were produced by using the PCR or polymerase chain reaction, and we have discussed the detailed account about the PCR or the polymerase chain reaction, what are the requirements, how the polymerase chain reaction is happening, right, and how the multiple copies of gene of interest is being created. Now in our today's class, we are going to deal with the step number 5, 6 and 7, that is how the recombinant DNA is transferred into cell or the host organism, then how the protein or how the culturing is being done of these host cells on the large scale. The large scale culturing is basically done in bioreactors. So we will basically learn about the bioreactors and its types, and then how the desired product which is being obtained by the gene of interest, whether it can be protein, it can be vaccine, how it is extracted and how it is manufactured in the for the marketing, that is basically the downstreaming process. So we are going to discuss these 3 steps today. Now when we learned about the host, when we studied tools, during that time I already told you that the host cells are made competent, and there were various methods by which the recombinant DNA was inserted into the host cell. The first was the heat shock, and the other were the micropipettes, microinjection, biolistics, transformations. So these are the methods that we have already discussed. These are the methods by which the recombinant DNA was introduced into the host cell. Now after the recombinant DNA is introduced into the host cell, then there is a selection made to find out the transformants and the non-transformants. The transformants are those host cells which have accepted the gene of interest, and we will only take up the transformants for the further processes, right. The non-transformants are removed because they are not having the gene of interest in them, and we select the transformants and non-transformants by using the selectable markers, which is the ampicillin resistant genes and the tetracycline resistant genes. Okay. So now the next step is how you are going to obtain the foreign gene product. First we will discuss it on the small scale, and then we will come on the large scale. So during the recombinant DNA technology, the main aim is to obtain the desirable protein. The protein which is produced due to expression of recombinant DNA in host is called recombinant protein. Now the cells which have the gene of interest can be grown on small scale also in lab, and the product can be extracted and purified. The cells can be multiplied in continuous culture where the used up medium is removed or drained out from one end, and the fresh medium is added from other end, so that the cells are in most physiological active state. So when the fresh medium will be added, the medium is going to have all the energy rich components that will support its division. So the physiological most active state is the lag phase or exponential phase. You remember children, we have learned about the growth curves, right, exponential growth curve and logistic growth curve in 11th standard. So we learned the sigmoid growth curve, where we divided the curve into lag phase, exponential phase and stationary phase, and the exponential phase was the most active phase, right. So the culture medium is supplemented so that the bacteria keep on dividing. Now this type of culturing produce large biomass, and as a result the yield is much more. Further, if you want to culture more, if the volume of the culture is 100 to 1000 liters, then the bioreactors are used. So here are the most common bioreactors. The diagrams are given here in the slide. These are showing you the bioreactors that are used when the culture is to be done for the 100 liter to 1000 liters of the medium, and specific products like enzymes, proteins are obtained from the plant, animal or human cells which are used as the host cells. Now this bioreactors is having all the optimum condition which are essential for obtaining the desired products. They have the proper temperature system, they have the pH systems, they have the salt, vitamins, oxygen supplement systems. All the conditions are basically in a way that it supports more and more growth. The most common type of bioreactor is stirring type of bioreactor. Now the stirring type of bioreactor is the A figure, where you will see there is a cylindrical curved base is there. This facilitates the mixing of content. In the center there is a stirrer, and that stirrer facilitates the even mixing on and the oxygen availability to the reactor. So whenever the stirrer moves, the oxygen is bubbled in the medium, and this stirrer helps in proper distribution of oxygen throughout the culture medium. The bioreactor are having the many systems like agitator system, which are basically used to churn the medium, oxygen delivery system, foam control system, then pH and temperature control system, and they have the sampling trays or the pots from where the culture mediums are removed periodically. The stirring type of bioreactor is two types, the simple stir type and the sparked stir tank bioreactor. So the first figure is the simple stirring type bioreactor, and the second figure is the sparked stir tank bioreactor. In the second type, the surface of the culture medium is increased by bubbling the air into the system. You can see here the oxygen is bubbled. In the B figure you can see that oxygen is added in the form of fountain, and this oxygen is bubbled timely. Why the oxygen is supplied? Because oxygen does the respiration, because all these cells are dividing and producing the more and more proteins, so they need energy. So for energy requirements they all require a lot of oxygen. Okay, so this is how the products are extracted by using the bioreactors, and after the products are obtained they undergo the last thing, that is the processing of that product, and so that it is ready for marketing to be sold out in the market, and that is why this is categorized under the downstreaming process. So basically the downstreaming process includes all those processes where the separation and purification of desired product is done, and during the downstreaming processes all the insoluble things are removed from the protein or the product that is obtained. Then there are certain preservatives or additives which are added, the labeling and the marketing is done, the price tags are put. So all these processes which make the product ready for final marketing and which is helping them to be a finished good is basically considered as the downstreaming processes. In case of drugs and medicine, the clinical trials are taken, otherwise they are directly marketed. So this is the complete process of your recombinant DNA technology. All the 7 steps we have discussed one by one. I hope you understood the concept. In case you have any difficulty, please call.